All right, welcome everyone to April 15th, 2025, Taxonomy Tuesday. Today we're going to talk about morels and similar looking mushrooms. We did get into it a little bit last week, but we're going to carry on this week. So I have a little bit of a presentation that I'm going to hopefully fly through, and then we'll get to looking at some finds that hopefully all of you are willing to share. So with that, I'm going to go over to this presentation here and share that. Okay, and let me see if I can get it into slideshow. Let's try that. All right. So morels and similar looking mushrooms. All right, the first thing up is that these are ascomycetes. So just in way of a brief overview, reminding people the difference between ascomycetes and basidiomycetes. On the left here, we have a drawing. Actually, both of these are drawings. And just as a reminder, the basidios have their spores coming off of basidia. The basidiospores are launched from these structures called basidia, and in the ascos, and here's a morel is the typical example in this artwork showing that the spores come out of asci, or a single one being an ascus, and there's typically 8 or multiples of 8 spores in there. So I kind of like this drawing and thought I would share that. So let's get into it here now. So morels are considered one of the foolproof 4 mushrooms, but are they actually so foolproof? There's a lot of people out there looking for them now, and I think it's a good thing that every year we spend a little bit of time talking about morels and some of the other ones that are similar to them that we don't recommend people collect for the pot. So this is the cover of National Geographic. Was the issue from April of 2024. So a year ago, there was quite an extensive coverage in National Geographic about morels. So it's a pretty good read. If you haven't seen that, go ahead and try to hunt down a copy. I think people would enjoy that. So I want to get rid of something that keeps popping up here. Let's put that over there. Okay. All right. So I did just a little bit of work on INAT just to see how many species of different things there were in New Jersey. So just quickly here, there's 4 different species of Morchella, the true morels, in New Jersey, and you can sort of see the distribution. Don't seem to have any being posted down in the Pine Barrens or in coastal Jersey, which is kind of interesting. So there's 4 species according to INAT that are found, and hopefully we'll see some from observations that people have. Americana being the most common, and punctipes apparently being the least frequently observed. I think the only one really that I've ever seen, or most commonly have seen, is diminutiva, and sometimes americana, but I don't see very many. So Verpa, and last week I think Hunter, you had some Verpa that you shared, you and Dave. There are 2 species of Verpa. in this rectangle, this northeast, but interestingly, there were none showing up in INAT in New Jersey. So Verpa conica, conic morel, or sometimes I think it's called the smooth morel or the smooth thimble morel. One was found yesterday. You can see the seasonality of this, which I thought was interesting. They should be peaking right about now in our area, if there are any in our area. Verpa bohemica, one found today, also peaking right about now. Then we'll move over to, whoops, what happened? Lost my slideshow. Well, hang on a second, folks. Somehow my PowerPoint closed on me. Let me pause the share. I guess it stopped altogether. All right, hang on.
Does this Verpa slide link to INAT where you saw that by any chance?
No, I don't think I
Linked it, that's okay. Maybe
set
later
it up that way.
we can look at it more later.
What is going on here? No. All right, everybody, sorry about that. We're back. Thanks, Jason, for presenting this, and I'll talk through. So Morchella, Verpa, now Gyromitra. Just wanted to show here just a few observations of this. I think in part they're so few because they're now being called other things. Some of them are being called this Maublancomyces. So we can just go to the next slide, which I think is a little bit about Alden Dirks here. I wanted to put this out there for reference. Dave has mentioned it. There's been some recent work that Alden and colleagues have been doing regarding toxin production, the Gyromitra toxin, and studying that, and that has led to some taxonomic overhauls and changes. So there's a couple of resources here that people may be interested in, and maybe we'll come back around and talk about toxins at a future session. But he has a kind of a short version of his paper, the full paper, and then he also did recently this year a NAMA lecture on the so-called false morels, but instead he's calling them Lorchels, saying it makes more sense to call them what they are and not what they aren't. So I put that in there for some reference, and also to draw attention that, you know, we love to eat morels, but we've got to be careful, because if you mess up and end up eating one of these Lorchels instead, you could be going down a path to get very sick or worse. Okay, so next, just a kind of a prototypical example of what one of these Lorchels or Gyromitra that we may have seen look like. This very brainy looking structure, which is very different from the true morels, which are more pitted. So to me, they don't look similar, but some people that are new to this could confuse them. And as you'll see on the, based on these observations in INAT, they peak basically in May, so they have an overlap. Next. So this new name, we talked about it last week, also this Maublancomyces korfii. There are a number of them that have been found in New Jersey and posted. So 10 observations, most of them in the northern half of the state. Next. Here's just a picture of korfii and its particular seasonality graph. Again, one was found yesterday. So people are finding these sorts of things right now. Next. Helvella, another genus that sometimes gets thrown in here with being something that looks a little bit similar to the morels. And quite a lot of species of these and a fair number of observations in New Jersey proper. And this is more universally distributed across the state, this genus. So we can look at the next slide. Here are the 8. And from some quick research that I did, most of these are typically found, more commonly found in the June to October timeframe in New Jersey, with one exception being that Helvella astabula, and that does show up in April and May. So people may be finding that so-called vinegar cup if they go out looking for morels right now. And next, how to find morels. So I put together the next 2 slides. These are all links to the Learn Your Land YouTube channel, which is put out by Adam Harriton. And there's over 2 hours, something like 2.5 hours of videos out there, which are very well done. And I encourage people to go and check those out. So he may even have more, but these are the ones that I went through and found doing a little sleuthing for this back in March. So good stuff. So with that, let's start looking at some finds that people have made here on the call. And just a reminder to have fun, but watch out for the ticks. Thank you. All right.
Isla, ticks have been bad for me already.
Yeah, I've had one myself already too. So, okay, so let me go to the email and I'll get looking at that. In the meantime, if somebody has something that they want to share from their own desktop, we can do that too. See what's going on in the chat. All right. So if anybody does have something, please put your name in the chat and we will circle back around to you. In the meantime, let me go over to the mail now and we'll look at these in the order that they came. And I'll see if I can share. All right, try to share again.
Lila, I put a link in the chat to a different kind of false morel that comes up in the late summer.
Okay. Do you want me to start with that first?
If you want to. Oh, okay. Well, no, we've got this thing up now. So this I'm glad Igor is here because I want to see what he's going to say about this. In fact, I sent Igor a detailed sort of account of the analysis that has been done with these so far. So I haven't seen any of these yet this year, but it's been a chilly spring so far. So I would be surprised if any would have come up so far. I checked today, there were none. So there's at least one species of morel that's growing on my property where I'm almost certain they're the result of a morel grow kit that I planted maybe around 2005. And in 2020, they started coming up. That was the first I saw, and this was the first one. And this was sequenced by this one was sequenced by Steve Russell. And there are two names that seem to apply, either Morchella laurentiana or Morchella norvengiensis. Those are both new species that were documented around 2016, I believe. There's a paper that's available. I'm not sure if I put a link to that paper in this observation, but I know on at least one of my observations I posted a link to the paper. But you can see here there's sequence for this. Actually, Steve posted 3 versions of ITS that he did, and so the problem here is that this is a morel from Morchella section distantes, and ITS does not do a very good job of delineating species of Morchella other than letting you know what section it's in. Except this one, or it might actually, there's another one from the same location, same time of year. And when I say same location, I'm talking 4 square meters. So it's a very small area where I planted part of this kit. So I did some microscopy. I think that last one, I think those were elements on the sterile ridge. And these elements are mainly just cylindric, maybe a little bit inflated here and there. The spores, there's a measurement put here. I think I remember the spores. That's the spot right there. That's where they grow. I planted them by this old apple tree on our property. That's where I planted the kit, and it's really a very small area in front of the apple tree where they've been coming out. So 5 years in a row. This, if they come out this year, that'll be 6. So I'm waiting to see, but I think it's going to have to warm up a little bit more. It's supposed to be in the 70s over the weekend, and there's not supposed to be any dreadfully cold weather in between. We had about a week ago, we had it was like 20 degrees here 2 mornings in a row, so that pushed things back, I'm sure. So I'm not sure if it's this one or the other one that was sequenced by Fundus. That I also have a link to that particular mushroom. Each of these first 2 years, there was only one fruit body. Here's the one from 2020, the first year that these appeared, and this one, I believe, the best match, the ITS, and you can even click on the link in the right hand column down the bottom here. You have to go way down. See ITS. If you just click on any of those blasts, just click on a blast. And I know, Igor, you said that this doesn't work very well, but they fixed it. So now if you scroll down to the bottom here, you'll see blast again. There. Now click on that blast, and it's going to take a few seconds for it to show the matches. So these are going to be matches with ITS sequences that are posted on GenBank. And I don't know if Steve, I don't think Steve posted these on GenBank, but my other one from Fundus that Igor helped with, that one, with the editing and so forth of that sequence. That one's posted on Fundus. That one's posted on GenBank, and that's going to be our biggest match. So if you scroll down now, you'll start to see the matches. There we are. So actually, it looks like, so you see all these names here, right? These are all things from section distantes, and you'll see here down Morchella Laurentiana. It's down here about that one. So Steve's sequence of ITS matched that at 99% cover, meaning the sequence NRRL, the one that represents this row, covered 99% of the sequence for my mushroom, and the identity was 99.59%. So that's the match. 99.59% of the base pairs matched. This is the type specimen for Morchella Laurentiana. So that's a pretty good match with the type specimen. And the other one that I've sent a link for was the one that Fundus did, and there's a 100% match for the type specimen of Laurentiana for that one. Now you see though these other ones, there's other ones that have a very high percentage of identity and 100% cover. So anything that's over 99%, you start to say, that could be the same species. But look at all the different names here: angusticeps, norvegiensis, purpurascens, brunia, schneideri, and some of these might be the names might be wrong. Okay, so some of these purpurascens might be one of these two new species. It's hard to say. But anyway, that's what you do when you get a sequence. You blast it on GenBank. And and by the way, these names up here, Eohespera was what was for a short period of time was a name that was later changed to Norvegiensis, because Norvegiensis was published before Eohespera. So anyway, that's just a little sort of window into what you do when you have a sequence, and you can blast it on GenBank. So if you go back now, just take your arrows back to this observation, and you might have to actually click out of this window. I forget how this works.
Let me close. I'll try
Oh
close.
yeah, just close that window and it'll go back to Mushroom Observer. Yeah, there we go. So now, if you go to the next observation that I sent, that's going to be from the same exact spot. This one was sequenced by Fundus. And this one, Igor helped me out with this. He was on my team. My team was Igor and Professor, jeez, starting to forget so many things.
Is it in here someplace, Dave?
No, I'll think of it though. But there's another sequence down here. So you can do the blast thing again. See where it says ITS? Yeah, just do a blast. Either one of those is fine because one of them's edited, but you get the same results pretty much when you do the blast. And you'll see here this time we're going to get a 100% match with the type specimen for Laurentiana. So that'll take a minute. You can go down there now. It'll show up now. It's not going to be shown. Oh, by the way, the Laurentiana here that's the one that's right on top. That's the one that we just looked at because that one's on GenBank, so it's being matched with itself. So that doesn't count. But if you go down further, you're going to find here Morchella Laurentiana down here, a little lower. There, that one. No, not that. Not Elata. Up that
Right.
one. 99% cover, but that's a 100% identity match. And that is with the type specimen for Laurentiana. And I'm pretty sure that this one that's right above it, that's called KOD 215A, I think that one's also Laurentiana before it had a name. That's also a 100% match. So now look though, also there's Norvegiensis up here. Norvegiensis, look at the one that's a few steps up, 100% cover, 99.72% identity. So that's a pretty strong match. But that's mainly because ITS does not delineate within section distantes of genus Morchella. It doesn't delineate very well. Although I have to say that getting a 100% identity match with the type specimen, that seems like pretty good evidence. But wait, there's more. Laurentiana is supposed to be a very early occurring species of morel, one of the first ones. Although if you go back to the observation now, you're going to see the date is pretty late April. 425. That's when this first showed up. The first time I saw this mushroom was the 25th of April. That's pretty late for a black morel to start up. Black morel is like the common name for morels from section distantes. So Laurentiana is supposed to be one of the first ones to show up. Norvegiensis is supposed to be one of the last ones to show up. So that sort of indicates that this could be Norvegiensis. But wait, there's more. If you go now to the next observation, which is going to be from a different year, I think last year. Yes, this is last year. See, this one's just starting to poke up through the little bit of like leafy debris and stuff. Look at the date on this, March 31st. Now, last year, 2024, was coming off of a very mild winter. There was practically no winter here in 23-24. So things did warm up a lot faster than usual. Our property is in a cold spot, so which could account for the late fruitings of those other two that we saw at first, which were both after April 20th. But this one, look how early it is. Now, unfortunately, I don't have a sequence for this one. And I think I might have some material that I dried. I'm not sure. I have to look around. But according to the authors of the paper that where they published Laurentiana and Eohespera, and Eohespera was later changed to Norvegiensis, which was predated it by a few decades. You're supposed to use RPB2. So that's a different region of the genome. So I'm going to look around and see if I have more material, in particular from this last year, from this very early fruiting. So these, by the way, these photos are going through a day-by-day a change in the morphology. So this was in situ. I left it there for for like 10 days or so. And twice a day, I'd go in the morning, I'd go in the afternoon and take photos of it to see how it progressed. So this progression is according to time, right? So each photo is a little bit later than the last one. So that's it for that last one. We're back to the beginning now. But the next observation is another morel from the same fruiting. So this is the last of 4 that I sent for this. So this is what this looks like when it gets mature, when it's completely mature. Look at this really deeply wrinkled and stalk, and it's very mealy also. The stalk surface is very mealy. I think I have a close-up of that, one of the photos. So I finally harvested this. Now that's not the same morel as the other one, but it was from the same fruiting. So the date is different. I think I didn't see this one until the 19th of April. but it was kind of hidden under some leaves, so maybe that's why you didn't see it. So, but given the fact that these two, this year, 2024, these came out so much later, or I'm sorry, earlier than the other years, I sort of have to wonder, did the place that sold this morel grow kit, did they put more than one species into the grow kit? Well, there's only going to be one way to find out, and that's I'm going to have to get more material and find someone to sequence RPB2. So you can see here, see how mealy the stock is? That's, I don't know how telling that is, because angusticeps gets like that also, if you allow it to become overly mature. Angusticeps is our most common black morel, that I find in the woods around here, and I've also found in New Jersey. So it's
When
a
you
very
say
interesting.
mealy, like, what do you mean?
We'll go one more photo. I think the next one shows it even better. Ah, there we go. See these,
Ah,
like, yeah, see, that's the stalk. It's covered with these mealy sort of flaky things.
that's
Um,
very interesting.
Is
Yeah,
that
yeah, and
called
justiceps
punctate,
gets like that also. I'm
punctate
sorry.
or pun, punctate?
Yeah, punctate. You can say punctate, but boy, these flakes are really big. Punctate is more. That's usually means like sort of smaller projections, like pointed sort of.
Okay.
So I would call this mealy.
that's
And
interesting.
justiceps gets like this also if you let it get really old. There's um, there's a few other close-ups. There's a ridge there's another that ridge is kind of dried out, and I think it's lost some of its character. But I think the next photo, that's inside one of the pits. So it's a little bit pruinose or finely tomentose. Um, so I've oh, here we are. There's the ridge. See the ridge? The ridge is actually kind of grainy. Um, so anyway, I made a list of all these different traits, went through the description of Laurentiana and Norvegiensis, which the authors of the paper readily admit are very similar. They're very similar looking mushrooms. Um, and I can't tell. I think there are a few more traits that seem to match Laurentiana a little bit better, but there are some traits that seem to match Norvegiensis a little bit better. So I don't, maybe I've got two different species growing here. The only way I'm going to figure this out is to get more material and find someone to sequence RPB2. So, anybody have anything they want to add or a question or whatever?
Yeah, I just want to clarify a few things. You threw in a lot of information, Dave, and I mean, I got all of it, and I got your email, and actually I've done some reading. I just didn't put it into words. But ITS actually is not that bad at resolving um, morels. You need to build trees. It's not superior to RPB or TEF. It's worse, but it's not terrible. I mean, you get a lot of resolution for many taxa, but there's going to be there are going to be a number of taxa or species that essentially share the same ITS, and therefore ITS lacks resolution for those complexes. And Norwegiensis and Laurentiana seem to be one of such situations. Now, you saw the sequence of Laurentiana, and I think that's a full length one. I couldn't find a full length sequence of the type of Norwegiensis, but I found a partial epitype sequence, apparently from the same location. And when I overlaid the two, they were essentially the same. So the paper that describes Laurentiana and or has Laurentiana and Norwegiensis in the same publication is a paper from 2016. The tree that shows that Norwegiensis has the same sequence as a bunch of other species is in an earlier paper by Richard, 2015. Now, a paper from 2022 by a number of authors. I think the lead author is a mycologist by the name of Loisides. That paper says that Norwegiensis and Lawrenceana are undisputed taxa, that they are actually different from each other and their status is undisputed. But they show a tree, a multilocus tree, where these two things sit far apart from each other, far enough that you say that they are different. So I agree with you with your email. You need to step away from ITS and you need to sequence either RPB 2 or 1, whatever is better, or TEF1 to get the ID for your species. For your morel that you planted 20 years ago under the apple tree. Another thing that I wanted to say that the ITS sequence from Fundus, I did not work on that sequence because I found it to be handicapped, and I refused to work on it because whatever was done to it was pure conjecture. I do not believe in reference sequences. I think it's cheating. I think it's unscientific. And I have nothing to do with that sequence.
Okay, I'm sorry. I thought that was one you may have helped with. Okay, so that was one you did not want to help with.
Yeah,
And
I
it was
dropped it.
Jean.
Yeah.
Jean, what's her last name?
Gene Lodge.
Lodge. Yeah, it was Jean Lodge. She did the work on that.
Right. Now, the one by Steve Russell, I believe that's trustworthy because he obtained it from nanopore sequencing. And that method, basically, yeah, I mean, it's reliable and it gives you, instead of a chromatogram, it just gives you the actual sequence right away because the computer puts the pieces together and just spits out the entire FASTA sequence at you. So it's a different processing scheme and different methodology for sequencing as well, by the way. So I found that sequence to be trustworthy. But the result is essentially the same. It matches Lawrenceana and it's going to match Norwegiensis. Now, Norwegiensis actually is a widespread, ubiquitous species, whereas Lawrenceana is parochial. I think it was only found in Labrador and nowhere else, unless we have now more evidence that the species is found elsewhere. But you need those TEF1 and RPB sequences, not ITS.
Yeah, the authors of the 2016 paper say RPB2 is what you should
RPB2.
use.
Yeah.
Yeah.
Okay.
Also,
So
yes, I know this one is Laurentiana is supposed to be parochial, meaning it's not known from very many places. But if it was put into a kit and planted, then it might show up someplace where it wouldn't, where the likelihood would be otherwise very, very, very small.
yeah, but your kid has to come from presumably where Lawrenceana was first
Yeah,
found.
I tried to find out where the kit was from, and I realized like yesterday morning that I think that my brother bought me the kit as a present, like a Christmas present. And I talked to him yesterday, but he doesn't remember. And I looked through all my financial records, like old checkbooks and things and credit card records, and tried to find out where I bought this from. But you know what? Most of these kits come from the Northwest. Most of these things, if I don't think this one was from Fungi perfecti, but most of these kits are sold from places in like Oregon and Washington and Northern California.
Right.
So
So it's probably local, but Norwegiensis, formerly Eohisphaera, is found globally. It's found in China, it's found in Europe, it's found in Eastern USA and Western USA. So it's
yeah,
very
but
widely, it's cosmopolitan. Okay.
yeah, but there's a strike against Norvegiensis in that the habitat is supposed to be calcareous. Is that how you say it? Right? It's the pH of the soil is supposed to be kind of high.
Calcareous.
Limestone areas. That is not the case on our property or anywhere, hardly anywhere in Northeast PA. There's some limestone outcropping near Stroudsburg, but that's like the only place I know. Anyway, so the habitat kind of favors Laurentiana, open areas, Both species, by the way, are reported as possibly associating with malus, right, which is apple. And that's why I planted this by this apple tree. By the way, there's also shagbark hickory not far away, but that likely has nothing to do with this. So anyway, yes, I think we can't say what this is yet. So there it is. Morchella section distantes. That's as good as it gets for now. Hopefully, more of these are going to come up, and hopefully there's. Well, I don't know if I want to say hopefully. Maybe there's two different species, but I kind of doubt it. But
I think it's weather, Dave. I think it's weather and moisture because it's.
Yeah, I think so too. I believe so
Yeah.
also. But I can't say that that's not true. I
Right.
think what they do in these kits is they probably put sclerotia in them. So if a vendor wanted to increase the probability that the kit was actually going to work, if they had sclerotia from two different species, they might put more than one kind in the same kit. I mean, that doesn't seem like a bad idea, really. But anyway, hopefully there's going to be some more this year. I'm expecting maybe by Monday, after we get a few days in the 70s. And by the way, I'm going to check my local spot for angusticeps tomorrow, because we had 2 days in a row that were fairly warm. Yesterday was up near 70. Today was in the mid 60s. There was some rain. It's going to get cold again, but not real cold. It's not going to go below freezing. At least that's what they're saying. But once they start, that's okay. If you have like a night that's in the mid 30s, it doesn't do any. It doesn't stop them. They'll keep coming out. But I'm going to check tomorrow. But anyway, thank you for your comments, Igor.
Yeah, I mean, I just want to sort of draw the line and say, my best guess, at least for the collections that were sequenced, with ITS, you only have two choices. It's either one or the other. I don't think it's anything else.
I agree with that. And there are more Norvegiensis sequences available in GenBank. There's only
it's from everywhere.
Yeah.
I mean, the original collection, the holotype grew in some mixed environment amongst rotting or decomposing, I don't know, some paper refuse. But I've seen collections from a Norwegian paper in 2020 that basically talks about Norwegiensis or morels from Norway. It grows in a variety of habitats, and I think there were collections in there, at least one that grew with apple trees. So this thing apparently is maybe not even mycorrhizal, and it can grow anywhere. And types of soil, maybe it likes acidic soils as well, or it can grow there.
Maybe, but that's not, the authors of the paper that I have, they were very specific about the habitat being sort of a high pH habitat.
Well, maybe refer to the type collection, or maybe just a couple of collections, but you cannot.
That they might. Well,
you have to explore.
Referenced a lot of collections in their paper, though.
Yeah, but the thing is, they have a lot of collections of different species, but you have to say examine maybe, I don't know, 15 or 20 collections of the same species and find out where they came from, and to have a judgment about the kind of habitat that allows for this mushroom to grow. I think Norwegiensis, because it's so cosmopolitan, it's very flexible. So my bet is that it can grow on acidic soils, but maybe it's not its preferred environment. But it can probably still grow on neutral or acidic soils. That's my best guess.
All right, let's move on.
Yeah,
let's move on.
So here's a different. This one used to be Gyromitra, and this is from New Jersey. This one was from Stevens State Park, northern New Jersey. And this is now Neo-Gyromitra brunea. Now, this one in older field guides is listed as toxic, and there are in some field guides there are descriptions of like people getting really bad headaches from eating these. And so I've never tried eating these, but Alden Dirk says that Gyromitra brunia is like an excellent edible, and he's one of his favorites, he says. But I'm still a little bit scared to try to eat it because just after having read in field guides so many times um, that it's toxic. But one thing seems pretty clear, that this species at least does not contain gyrometrin. So, or if it does, it's in trace amounts. So what makes this different from korfii? Well, this one has like really sharp folds, like these creases that are really abrupt. And look at the top of the fungus is actually looks like two discs that are sort of pushed together, because there's like this, it's not a crease on top, it's like a separation actually. And that goes right across the entire long sort of arc. You can't see it in the photo very well. But I think, I'm not sure if I did microscopy on this. There, you can see this one. You can see this. See how those folds are really, they're so sharp sometimes that they actually sort of break apart. Well, they don't break, they just grow like as two different discs, pushed together sort of. But you can see here that this one looks a little bit different than whatever that new genus is for korfii. I can't remember how to say it. Blancomyces. Well, Blancomyces, right. Yes. Yeah, this one was, I found this species at Stevens once or twice on other times as well. So if you click on the little thing up top here, yeah, and then click on show original image or whatever, that's going to come up somewhere. You'll see it on the screen somewhere. Show original. It's usually like in the lower
Yeah, I
left.
got it. This is it.
Yeah, and okay, there. Click on the see, it's got a plus. Just click on the photo now; it'll enlarge. Okay, no knobs. There are no knobs on the poles of the spores. No knobs. Now, if this photo doesn't really capture it, you can't really see it, but there is ornamentation on the walls of the spores for this species, and it's like these clouds, these really thin clouds. You can almost see it in a few of them on the lower left. And you only see that real faint ornamentation if you mount in H2O. So if you mount in KOH, it destroys the ornamentation. It just dissolves it. So it's just probably like this like mucus sort of stuff that's clinging to the walls of the exterior. But the main thing is, on the poles, you don't see knobs. I forgot how to say it again.
Maublancomyces.
Maublancomyces korfii has a conical knob, a really big conical knob, like several microns long on each pole. And so that is one way that you can definitely tell the difference between Maublancomyces korfii and Gyromitra brunia. In this same location, Stevens, I believe I've also found Gyromitra caroliniana. I think that's what it's called. The one they call Big Red.
Yeah.
Yeah. A long time ago, I saw it there, like 1998 or something like that. But I haven't seen it there since. And I'm pretty sure that I remember that because it was big and it was red. So I'm pretty sure that's what I had seen there.
Can we zoom in on these? Can we zoom in on these spores again? Looking at that clouding at the.
Yeah, it's hard to see. You can see it a little bit here and there, like little sort of appendages here and there on the perimeter, like along the spore walls. Yeah, a little bit here and there. You see, yeah, and some of them are really thin. They're like thin and sort of longer than they are protruding.
Yeah. Okay. Yeah. Now
if
I
you've
see.
got a good scope, if my camera didn't pick it up, okay. But if you've got a good scope, you mount these spores, and you should get spores from a drop. And sometimes it's hard to get a drop from these things because they mature slowly. And there's been times I've got no drop out of these more than one of these kinds of false morels. But if you get a spore drop, you've got mature spores. If you mount them in H2O, you will be able to see those little clouds along the exterior. And you know who's got a good photo of that is Michael Quo on Mushroom Expert. And he probably still calls it Gyromitra brunea. So I haven't tried eating this one. I have eaten Gyromitra Maublancomyces korfii. I have eaten that one. At least I think that's what it was. It could have been Maublancomyces americana gigas, which is virtually identical. Even the spores are almost the same size. You probably have to get a material sequence to tell the difference between korfii and americana gigas. But anyway, there they are in the ascus. It looks like you can count 1, 2, 3, 4. Yeah, you can actually see all 8 spores inside this ascus. Sometimes you can't see all 8 because you'll see an ascus and 1 or 2 of the spores will have been expelled. But you can see them all in this photo. So, okay, I think that's pretty much all I got. There is another thing that I sent as a link. I think I also sent this other thing that we could look at, but that can wait. But the thing I sent in the link, that would be pretty cool to see. Because it's a false morel that grows in fall. Well, late summer. I don't know if you get it in New Jersey. If you do get it in New Jersey, Stokes would probably be the most likely place to get this. So there's 2 of them. There's it's Paragyromitra infula, and there's also Paragyromitra ambigua. It looks very similar to this. I think there might be a difference in spore size. I forget, but this is the saddle-shaped false morel. It grows on wood. I think usually conifer wood. And you can see the date here is 10/29. So this one was well into the fall. I found this. I have found this in New York State earlier in the season, like in August. And like I said, if you were to find this in New Jersey, I think Stokes might be the best bet. So this one should not be eaten. I forget what Alden said about this one. I think he said that.
It contains gyrometrin.
Excuse me, Hunt?
It contains gyrometrin, right?
No, I think he said that it doesn't. Or if it does, it's a very small amount. But he thinks there might be something else in it that's bad. It has a bad reputation. These have a bad reputation as ones that will make you sick. So the spores
found those in northern Wisconsin in like July or August, like you said. Pretty interesting. Yeah. In conifer.
Yep, no ornamentation on these spores either. But they do have consistently 2 oil drops. So that would be described as bigutulate, meaning basically 2 gutules.
Fine. Interesting. Thank you.
Yeah, very interesting stuff there, Dave.
Oh, sure. Yep, glad to share. You're welcome.
All right. So I think Hunter sent some things, so let's open these up. I haven't had a chance to look at these yet.
Yeah, and I only threw some photos together just earlier today. Things have been really crazy for me. So I just did just to make sure we had enough photos. So if there's other people who have stuff and want to go, I can wait too. So But they're morels too, mostly. They're morels, so we can run through them.
All right, am I not sharing? Let me share here.
Yeah, I'm not seeing anything.
Okay, share.
And some of these I'm pretty sure I shared last year. This is Morchella prava. And this is kind of a not that common morel that has very dark pits, and which make it look like a young yellow morel, a young americana or another species, but it maintains that darkness as it gets older. And I think it's a really nice looking morel. And so this is just another one that we have in North America, but not so common. I don't know if Dave has anything to add on that. But just thought I'd put it out there, keep your eyes out for it. I think.
What's the habitat? Because I'm pretty sure I found this species years back, 20, 25, 30 years ago. The apple tree where I used to find them has been dead for 25 years.
Okay, this was a dying elm along a creek. So, but there were cottonwoods around too, and but pretty typical dead elm, I'm pretty sure. So Creekside, Central Illinois.
Do you ever mistake sweet gum burrs for these?
Oh yeah, isn't that frustrating? And even
Yes.
even the sycamore seed pods, even those kind of throw me off sometimes. But yeah, we don't have a lot of sweet gum, so that only happens if I'm hunting in Southern Illinois. It's very, very rare that'll happen to me in Northern Illinois, and not so much in Central either. But yeah, those are I love sweet gum balls. They're so cool. Really, I mean, amazing natural beauty in those for sure.
Yeah, those ones with the dark pits, like you were just showing, those you can tell by what they look like. But there is another species in section Morchella, Morchella ulmaria, and I think you probably need to sequence to tell that one. I think I may have found it by elm, but I can't be sure. So I didn't send out any links to my observations because I honestly I can't tell. I would have to have it sequenced. And I think I may have sent it to somebody, but I didn't get any results.
Were you saying you think of Prava as growing with something else? Or
No, I think there's yet another species of Morchella. There's Prava,
no, I know.
americana, and then there's this one called Ulmaria.
Yeah.
And that one's different from the other two.
Okay, I'll have to look into that one. That's like UMA, Ulmaria.
ULMAR.
Oh, Ulmaria. Yeah, like elm. Right.
Yes.
Okay,
Yeah.
got it. Okay. Right. I have heard of that one, and I will look into that more. Yeah, cool. Thanks.
Just for the reference, Ulmaria, as the name suggests, grows with elm because ulmus is the genus of elm trees.
Yeah, and the ones that I have that kind of fit the description, which is not that different than Americana, is I find them near elm.
Yeah, and ITS resolves all three species, so you shouldn't be a problem.
Oh, really? Oh, good. I think I sent a sample to somebody, but I never got any feedback on that. I probably still have some here. That I'll send. If I do, I'll send it to somebody.
So I was thinking these are diminutiva, at least the bottom one. And I don't know if people disagree, but
No, I think so. What do you think, Igor? I think these are diminutiva.
Yeah, I mean, diminutiva, most likely.
The longer pits and
Yeah, the ridges are arranged not haphazardly as they are in Americana, but yeah, they line up almost like in the same fashion as the black morels. Less disorganized, more organized than Americana, and they're small, they're pale. Yeah, I think chances are this is diminutiva or maybe even Virginiana, except that Virginiana, or I should say Septiformis, that's the one from the south, or it probably doesn't reach Wisconsin
yeah, or Illinois. Yeah,
or Illinois. Yeah.
I am gonna, I sent one, like more email when we got started that I wanted you guys to look at to just make sure it's not Septiformis.
Septiformis.
Yeah,
If
but,
this is tulip poplars, this one only grows with tulip poplars as far as I know.
no, the last one I want to show you, it only grows, it was right next to a tulip poplar. It's probably just a, americana or something, but we'll get to it. What else we got, Lila? We can just kind of run through them. So this is a half-free Morchella punctipes. And so it has that mealy look that Dave described, I think, all the time. And that's one of, had learned that's how it got its name, punctipes, because it is punctate on the stipe. And then these half-free morels the cap attaches halfway up the cap to the stem, as opposed to these other morels that we've been looking at where they attach at the base of the cap to the stem. So Verpas, those attach at the very top of the cap to the stem. But these half-free attach halfway. And so the pits are pretty linear on this one too, but their morphology can vary. So that's Morchella punctipes. And then these are the biggest morels I've ever found. I thought people might get a kick out of just seeing them because they're just really big.
Monsters.
Yes, and they do get bigger, and people have found the bigger still, but this was amazing to to find these so large, and I thought you'd get a kick out of it. So probably Morchella americana, I think.
Yeah, that's great. That's a few meals there.
Yes. Yeah. And then this is Morchella angusticeps, and this is kind of the most common black morel in Eastern North America. I showed it to you last year, and I really never got into black morels till last year. We found over 100 of these, and it was cool because they were growing in a spot that I had pointed out like 3 years prior as prime morel territory, but didn't see anything. And then boom, sure enough, the spot produced a lot. And so that was kind of neat to be able to recognize some good territory and then have that confirmed. And then, so this is a Verpa conica. I had showed one last week that hadn't opened completely. But here is this Verpa, and the cap, it just attaches at the very top and separates easily from the stem.
I think Steve Russell has proposed possibly more than one genetic species in the Morchella conica, or in the Verpa conica group.
Yeah, I would love to find some good specimens, and actually I do have, I have that one from last week. I should send it to him. Remember that one last week was like,
Oh, the one with the, it's a little
ambiguous?
bit wrinkled. Yeah.
Yeah, maybe I'll send that to him. That's a good idea. All right, and
So
then,
just so I'm seeing that last one right, the hymenium is on the outside top of the cap, right? But this thing looks pretty capitate sipitate, more or less.
The fertile surface is the cap surface. Correct.
yeah, but yes, it looks like you said, captate.
I think these have really big spores too. I know I've looked a few times. I'd have to double check that. I think they're like over 30 microns or something. Let me check.
Yeah, I've never seen anything like this in New Jersey, so I don't really have, anything to go on. I'm sure once I handle the thing, I could figure out that it's a ASCO, and yeah, the hymenium is on the top of the cap. But from this photo,
Yeah.
it just kind of looks like any kind of mushroom.
Yeah, yeah, the underside is pretty nondescript. Obviously no gills, obviously. So yeah, they're pretty early when I found them. So, like, yeah, you'll find them before finding real morels.
Yeah, the conica spores are like 22 or 26 by 12 to 16. The bohemica are the really big ones, 60 to 80 by whatever, 15 or 16, something like that.
Okay, that's what I was thinking. And I've actually never found that species, but I guess that's what was lodged in my memory was that the one kind of Verpa had really big spores.
Yeah, that's
Wow, 50,
bohemica.
50
Yeah,
to
but
80 though, that's huge.
yeah,
Yeah.
that's huge. These Verpa conica, the spores are uh, about the same size as most morel spores.
Yeah. The other thing about bohemica is they're 2-spored sacs, not 8 spores.
Ah, that's one reason why the
They're so
spores are
big.
so big.
Yeah, that's interesting. 2-spored. Yeah.
Yeah, if you want to get spores from one of these, you just lay it down on a slide. Just lay the cap down on a slide, the surface right up on the slide.
I hope I can find more to do that with this year, because as you guys know, I was traveling with that other one, and it still looks great. It's in the refrigerator, but I just don't think it's gonna leave spores now.
Yeah.
I think sometimes after I put a mushroom in the refrigerator, it goes dormant. It
Yeah,
doesn't produce spores anymore.
right.
At least
Okay.
sometimes when
Well, maybe I can still
I've done that.
send it, maybe, dry it and send it in, maybe. Maybe. I mean, it does look great, like, do you think I could?
Oh, get spores out of this one?
No, dry this, chilled.
One that she's got in the fridge for a while, dry it out and send it off for DNA.
Oh yeah, it should be all right.
Okay, yeah, it looks great. Okay, good. Yeah, and I have a little, I think it might be a bohemica, but it's hard to tell those apart from the punctipes sometimes, I think. The half free.
Oh yeah, I've seen observations on Mushroom Observer where I would get involved in the discussion, and we couldn't decide.
Yeah,
Just based on what it looked like. Was it Verpa bohemica or was it Morchella? Well, actually, this one of these discussions was out west, and I think that's where they still have semi-libera, or they might have some other half-free morel with yet another name. I forget. The old name, by the way, is semi-libera, which means half-free, but that's a European species. They might have it on the west
oh, that's
coast. I'm
cool.
not sure.
I had not heard that. That's neat. I like it. So this is probably maybe a Pizzazza, but I thought it could be a Dicinia or the pseudo-Dicinia. Is that a new one?
I think it's Piscidicia or something like that.
Oh, yeah.
Piscidinia,
Pis,
I think. This looks like I would bet that this is Pisicepharium.
okay. I have another one. I think it might be next. That is, yeah, this is a Lorchel, would be at the broadest definition.
This one might be Disciotis venosa. I think that's still a good name. I don't find that around here.
That's in the group though, right?
I think, yeah.
Yeah.
I don't know if, well, it used to be considered in the group, but I don't know about the genetics, that it might not be on the same tree.
Okay.
But well, let me just review and, let me find a link to I think it's Disciotis venosa, something like that. I'll look on Mycological because they're kind of up to date, but they might not have it because they might not get it up there. Let me look. I'll put something in the chat.
Okay. It also looks a little bit like Piscidicia persicula or
Yeah, that's a relatively new species, and I think I may have found it in my yard, but I'm waiting to hear about sequencing because I think I sent it to Danny Miller.
Oh, yeah.
Anyway, yeah, let me look. Let me see
Okay,
if we can
also
get a page.
Pseudodysthenia melaleucoides looks a little similar to,
That one I don't think I know.
yeah, if the skin might be too thick on this one though, it was in Alden's, talk, but, yeah, and it's a pseudodysthenia. That's a really good talk. If people haven't seen it, should be available on YouTube, and it's really good. And he puts the spores photos right next to the picture of the mushroom, or drawings of the spores. And so it's kind of cool. I'm really hoping to continue to work with it long term on these Lorchels. I find them really interesting. I'd really love to get them down. Should we move on and we can come back to that, or there?
Yeah, I just put a link to the mushroom expert page for Disciotis venosa.
Yeah, let's check that out if we can.
Yeah, and I'm gonna look and see now if there might be a new name for it. So I'll be back.
Okay. Oh, can we?
Yeah, go ahead and look at it.
Oh,
I'll
there
keep
it is.
my mic unmuted, but I'm gonna look on.
I wonder if that is the Pseudodysthenia melaleucoides.
Oh, maybe.
Yeah, maybe.
Well, I'm in a different window now. So that would be pseudodysthenia.
Yeah, and
Let me
then
just do a
okay. Yeah, the flash just on mine seems so thick, but of course that can change with conditions, and they flatten out and thin out with rain and stuff. Yeah, so maybe it is that.
What was the species name? Because I googled pseudodysthenia, but it's only giving me pseudodementia.
Oh, melaleucoides.
Mello, Mela or Mello, MELA or MELO?
Meleucoides.
Okay, I might not have this spelled correctly, but it should be close enough. Oh, no matches.
Huh.
Oh,
That's-
wait, no, there are results for pseudodiscus melaleucus. What's that? Oh, it says it says there's no matches. Let me
It's definitely in his presentation. I've got the screenshot with this spelled out. But I'm liking that picture of this right next to the morel there. That's pretty neat.
Yeah, and if you look at the spores too, it looks like they're pretty smooth. Or I'm not smooth, but there's no oil drops. And I think that probably sets, oh, and there's no apiculi either. It looks like on these spores. So that would set it apart from Dicina. So Dicina ancillus, which was Gyromitra ancillus, that one has apiculi, these knobs on the poles.
Yeah.
And they are I forget what they're shaped like. I think they're just kind of square or rounded. And then the other one, the Piscina Piscidicina, however you say it, that one has the fishtail apiculi.
Oh,
They're pretty distinct. I've
yeah.
seen this because I've scoped these once or twice, and the apiculi, they really do look like a fishtail.
Yes. It's like a two protrusions at the base, basically.
Yeah. There's like two projections on one apiculus.
Yes. Yeah, cool. Yeah, so another case of just needing to get to the microscope with these for sure. Well, it's close on one of the others. So what else do we have? Anything more, Lila, or oh yeah, so this is this one I found by the tulip poplar. For sure. And I was assuming it was Americana, but any chance it could be the Septra Septriformis?
Those are Americana, I
Yeah.
would say.
Okay.
Little
Cool.
May apple coming up here next to it.
Yeah, a little one. Yeah. Yeah, I saw a lot of the mayapple rust too, and I meant to take a photo, but I'll have one for us next week. I love
Okay, was this year or last year, some prior year?
This is just a couple of days ago.
A couple days ago, nice.
Yeah.
Yeah, cluster of them, wow.
Yeah, I found a few that way. Yeah, I mean, it's a pretty good season down in Southern Illinois, and yeah, they were coming out like in this fashion, more than once, which is really nice to see. Yeah, that's always good. True flush, yeah.
Great. Was that everything from
I
you?
think so, yeah.
Wait, here's one.
Oh,
Is
no,
that-
that's it.
That's just
That
a
was
note.
there,
Okay.
yeah.
All right. So let me go back and see if anybody else has put anything in. I don't see that anybody else has. So I'll make a call for anyone to share morel, something that looks like a morel or something else.
Can I share a map?
A map?
Uh-huh.
Sure, go ahead, Chris.
Right.
Hopefully it's a map to something good.
I just want to say I
might
put
be.
another link in on the chat for Mycological. They do have Disciotis venosa, so it's a current name apparently, or maybe it's been changed. Maybe Alden changed that one. I'm not sure. I would have to check.
So this is the map. What happens when you type it in INAT? Type in Morchella. And how it cuts right across New Jersey like that. that like perfect line there. Now I was looking for one, but a geologic map, but they want you to like pay for it in some way. But if you look at a geological map in New Jersey, there's almost an identical line.
Can you zoom in on what you mean or
You see how it just cuts across?
That whole lower third of the state is kind of devoid of them. Jason probably can tell us what the geology of the soil type. There is something I've seen it on maps too that runs right through there.
Oh.
Just for reference, can you point out where Interstate 78 and 80 are? Those run west to east.
They're up here.
78 and 80 is way up.
Yeah, Allentown is where 78 goes across. You can just see
Okay,
Allentown,
that's
and
right
then-
there.
Like almost where it
Yeah.
says New York.
Yeah.
Oh,
That's
that's where
where
80
80
is up
80.
there. Right.
Yeah.
Okay.
Yeah, I think
Yeah.
this is probably it right here, 80. Yeah.
Because that's where I used to find some morels in New
Yeah,
Jersey.
that's way
Relative to
way
the-
up. But if you zoom out like that line, the way that it just cuts right across like that, if you look up a geological map, so I guess that's my plan is be on the other side of that line.
I'm not sure if Jason's here or not. He's the one that knows about this, but that follows that exact line of inner outer coastal plain in South Jersey, and then maybe even devoid in what is it, the Piedmont? No, that leads into the Piedmont up in North Jersey. But Jason would speak on that if he could.
Yeah.
Yeah, and I mean, I think it's common for coastal areas to be like devoid, or like I know in Alabama, they don't find them south of like Birmingham at all. You gotta get above like Birmingham to start finding them. So in Florida, there's none, in the like lower part of the state. Even though they're known to like sandy soil. So yeah, it would be interesting to hear more. And then New York, it looked like they had some more coastal finds there.
Yeah,
Yeah.
I think those forests are just so different. You know, the trees that we get in South Jersey are odd. You know, they.
Is that
I-
where the Pine Barrens is?
Yeah.
Okay. So there aren't morels in the Pine Barrens really much, huh?
Not much. They
Not really.
are.
Acidic soil, no trees that can be mycorrhizal with. It's a morel desert.
Yeah, it's kind of like northern Wisconsin. There's really, I mean, not that it's anything like northern Wisconsin, but which is like so much more north, but they don't get much up there either.
There is a rogue apple tree here or there in the Pine Barrens that I'll find a morel with. And when I say a morel, like two, but not in abundance, you know, as with other trees in better settings.
I've never had luck with apple trees yet, not one yet ever.
Yeah, I haven't either. It's dead elms or dying elms, and they can even be small elms, like 3-4 inches in diameter.
Yes.
It doesn't have to be the great big elm. So, and of course the ash trees, but hard to find a living ash in New Jersey at this point. That isn't, 1 inch or smaller in diameter.
Yeah, same here. Yeah.
And because of that, lack of morel associated trees, I do have the best luck with tulip trees in that area on that map that we just looked at, where there are very few observations. I have pretty good luck at some pretty good spots but it's only tulip trees, and it is more or less on the edge of the inner outer coastal plain. So not quite the Pine Barrens, but very much Pine Barrens esque.
Yeah, the furthest off of that line that we found a single one, not last year, but the year before at Clayton Park, which is pretty far off of that line. And that park has big, giant old tulip poplars. But other than that, like that place is almost like a little island. Everywhere around it is completely different habitat.
Yeah.
Emily, where did you find your morel that were
was-
yet to sequence?
I was just going to bring that up. That was in
The fire pit morel.
Yeah, fire pit, right?
Yeah,
Yeah.
that's a fire pit. So here
Yeah.
in Vineland, New Jersey, in Cumberland County.
Yeah, so it's like in that area of the map where there are no morels on either.
Yeah, right.
Have you
No,
been keeping your eye on it, Emily?
I mean, I take a peek out there. I haven't really been out much, but I have been out in my yard and I haven't seen anything there. We also didn't have any fires last year, so maybe that has something to do with it.
Yeah, you couldn't legally.
Well,
Good
Right.
God,
Oh,
yeah.
right.
Emily has
Exactly.
a specimen, so I'm hopeful. I'm hoping to submit this for sequencing, either whatever place that we decide to do a bulk sequencing, or maybe I'll call up Linus Kuzma and he's going to do that sort of, as a kind of one sample, or send him like a couple of samples of my own. Yeah, but we should do this. It seems like a very interesting species.
Yeah, I agree. I still have it.
It was a
Excellent.
black, right?
Mm-hmm.
Yeah.
Yep, one little black.
So, you guys lost all these chestnut trees, right? Like historically, the area.
Yeah,
Yeah.
they're gone, just like
Yeah.
ash.
There's a rare chestnut here and there that I saw one in southern Illinois. I've seen one in central Illinois. I don't really see them here either. I was curious if morels would grow with them, or at what else would do chestnuts like, if anyone had knowledge of that.
I think a resource to answer that question might be McIlvaine's Field Guide, because in the 1800s there were still a lot of chestnut trees in North America.
Hmm,
You know what? I've got his book handy. I'm going to get it. I'll look.
okay, thanks. That'd be interesting. Oh, yeah.
There is, speaking of the chestnuts, there are there's I don't know, a few groves or something. Some researchers did find some in southern Ohio, and they've been busy cloning and grafting them and whatnot. And I think they might have found some in Indiana as well, but there was a fairly large disease-resistant grouping, and these are big. These are not the huge giant ones, but we're not talking saplings from what I understand that they found. So there's apparently some genetic diversity out there that
Yep.
are some that have survived.
Pennsylvania DCNR has been planting hybrid Asian American elms that they think might be more resistant, or chestnuts. I mean, chestnuts that they think might be more resistant. So we have some here, but they're not very big. I'm going to look up this to see if what McIlvaine says about where morels grow, because I think he may have mentioned chestnut.
There's a pool on the Mississippi in Wisconsin somewhere that like off that pool, there's supposedly a grove of the large elms that somehow were resistant to the Dutch elm disease. I'm dying to get there, but I think it's only approachable by water pretty much. But it is interesting.
Yeah, you do. You know, I see occasionally in New York State a random fairly large elm tree still, but it's
Yeah.
out in the middle of kind of nowhere. And I've always suspected that that isolation maybe in part was why it survived.
Yeah, it makes sense. And yeah, with this grove, you know, in kind of a isolated area.
Isolated area, yeah. All right, well, I think I'm going to stop the recording. Thank you, everybody.